Okay. So our next presentation is by Yuhai Tu from IBM T.J. Watson Research Center, and he's going to talk to us about thermodynamics of information processing in living systems, with part of it crossed out, the energy cost of timekeeping of a single cell.
All right. Oh, thank you for staying until the very last session. Okay, so so I'm going to tell you something along similar line as you just heard David. He's sort of talking about computation in general. I'm thinking about along the same line, but specifically for living systems, that how does it use energy to compute things, to do information processing, and in particular, I'm going to tell you a story about timekeeping. How does a single cell keep its various time rhythm in terms of keeping the biochemical oscillation goes on, for example, for circadian clocks and so on. So at the end of the day, I hope you can, I can answer you, I can convince you about this. The answer to this question is how many ATP per day is needed to run a biomolecule clock, okay, single molecule clock. All right. Okay, so physicists has been thinking about living systems for a long time, for as long as, you know, but probably the most famous one was sort of musing about living system is probably Schrödinger. Well, at least in my knowledge, that he wrote a book in 1944 called "What Is Life?" Right, sort of very big question that only a Nobel Prize winning physicist can ask. And in this book, he listed a bunch of his thoughts about what life really is. Many of those thoughts turn out to be, you know, let's just politely say partially wrong or partially correct, or just doesn't have enough information to. But there's one thing he said in this book which is absolutely correct. He said that, Schrödinger said in this book that life feeds on negative entropy. Of course, he said it in German, right? I don't know German, but anyway, this is the translation. And it's basically what he's saying is that life costs free energy. Why did he say that? It's pretty obvious, right? So if you let thermodynamics second law run its course, then you get into the most highest entropy state. You will not have order. Order, as we see in life, cannot exist without energy dissipation to maintain the order. So that's the question. That's the sort of a general conclusion he gets from his general observation. Okay, so this is the slides you may have seen yesterday. I mean, if you were here yesterday, you've seen this examples in living system. The central question for, you can see from all these particular systems in life, is that how do living systems process information accurately with noisy components and stochastic reactions? Noisy component because mostly because the number of molecules involved in this computation is rather small, and the reaction as a result is stochastic, and also we live in room temperature. This is a very, very high temperature as compared to low temperature physics, where the noise is small. And also take, for example, for a neuron, we heard about brains and so on. The neuron firing rate is actually very, very noisy, right? You don't fire, if you say I fired 100 hertz, you don't fire every 10 milliseconds. You fire as a prolonged process, which have a lot of noise in it. So the question is, as I said, was saying yesterday, that the first question is how? How does it do it? How does the various living system do this? I'll tell you what I mean this. And how much does it cost? That's the second question, energy cost for biological functions. Now, then we come back to a little bit to the theme of this conference. It's basically about information lens, information processing. And this kind of question of energy cost, as we heard from the last talk, has been asked before and has a sort of an answer in simple system and simple processes. And here, Ralph Landau, which you heard about from is our IBM former IBM colleague, here actually wrote a paper in this very obscure journal called IBM Journal Research and Development in 1961. I don't know how many of you know this journal. But anyway, the title of the paper is Irreversibility and Heat Generation in the Computing Process. In that paper, he basically outlined what's so-called the Landauer's principle. And this question was originally asked actually by people like John von Neumann. So in this erasure process, as we heard the last talk, energy cost, the minimum energy cost is kT log 2 because you actually change from, you basically erase two possibilities into one, right? So basically there's an entropy reduction, so which you need use energy to achieve. Okay. So can we ask the same question about information processing in biological systems? So that's the question we try to answer. Then we go back to living systems. So how is free energy in living system being used? So that's the question. So you open up a textbook and you find, okay, well, energy is obviously used to do some sort of mechanical work. Oops. Here you see a movie of a kinesin dragging this large ball, this cargo, on a microtubule, and this is burning ATP to drive this. So this is a motor, molecular motor. And you probably can imagine that to build these bonds, chemical bonds, like biological synthesis, you also need energy to build chemical bond. If you open up a textbook, let's say Bruce Albert's Molecular Biology of the Cell, he actually have a diagram here which says, well, okay, you harvest energy by metabolism, light, and so on and so forth, and you get ATP as a currency of this energy, right? So you have these ATP floating around. What do you use it for? Well, biological system, as I said, used to synthesis, transport, moving around, and you also do a lot of plumbing, pump and heat and so on. So in this whole category, all these categories of how energy is being used, he not mentioned one thing, which I think is very important. He missed one thing, which is information processing. Something like regulation, control, memory, computing in the living cell or larger, biological organism. You certainly some of the energy is being used to carry out those functions, which is actually critical, which is what makes living things living. Okay, so to understand the energy cost in those processes, in my mind, is actually probably more important than understanding the energy use in these processes. Okay, let's go back from biology to engineered system, right? So engineered system is designed to consume energy to carry out certain function or work, do work. Of course, we know how a motor engine works. But for it to work, you have to put in fuel, and the car will run. So energy is being used to do mechanical work. But you also can imagine that these days, that lot of energy is actually being used to do computing, right? So look at this computer, IBM Blue Gene chips and IBM Blue Gene supercomputer. And if you check out on the internet, this is a statistics from 2018. 5% of all energy consumption in the US goes just to running computers. 5% of total energy consumption. And it's increasing pretty fast, right? I guess before Turing's time, it was zero. Okay, so of course, corresponding to this sort of engineered machines, we also have biological machines, right? This is what a biological machine looks like. This is an engine. This is an engine for bacteria to swim. This is really a rotary motor here at the bottom of this very, very long flagella that it rotates, that propel the cell swimming forward and changing direction. And you also have a corresponding so-called information processor or computing machine in living system. These are these biochemical networks. These are made out of proteins, and they interact with each other through biochemical reactions. Or in a larger organism with multicellular organism like the brain, you have many, many, many neurons, billions of neurons interacting with each other with synapses, and gap junction, and give rise to higher level computing and information processing capabilities. Okay, all right. So let's go to the subject of this talk. Is basically what is the thermodynamics? Can we use thermodynamics try to understand energetics in living system? Right? How does it use energy to carry out certain function? So I stress that you don't just use energy for anything. We're interested in trying to understand how energy is being used to do some just higher level function, right? Higher level means that it's doing something that it's not sort of intuitive, like using energy to move and so on. So the first example, which I also do yesterday in my intro, is that in '74, Jack Nino and basically this is a picture of John Hopfield and simultaneously Jack Ninos independently found this so-called kinetic proof reading scheme for biosynthesis, not just for DNA replication, but also for protein synthesis in the ribosome. Okay, basically what he's saying that, okay, if you do this based on a template, coupling a template, then what you find is that your error rate will be this P. P can be very small because you could have a 10 to the minus, because the energy difference between the correct base pair and incorrect base pair, you could have a small, relatively small 10 to the minus 3 error rate, but that's not good enough. So in order to improve from 10 to the minus 3 to 10 to the minus 6, how can you do it? So he figured out a mechanism to do it by basically what Charlie have told you before in the intro, that actually basically you basically once in a while after you synthesize the next base pair, you also retract it, right? By doing this with some frequency, you actually can make sure that you actually check in every base pair once in a while to make sure that you can go from 10 to the minus 3 to 10 to the minus 6. All right, so that was actually a very good general mechanism algorithm in living system to do error correction. And you also seen this picture I put up yesterday that actually Charlie then thought about, and Charlie talked about this too in '79, how this particular mechanism or this type of error correction mechanism based on a chemical reaction, how much energy would it cost, right? And how does it relate to the error that you're actually able to improve? All right, so that's just the early example, right? This is 74, 79. What about now? So, okay, so we try to take this spirit. Of course, the advantage that we have now is that we know a much more than about biological system than in the 70s. So we have a lot more example for us to use this concept to actually try to understand whether how energy is being used to improve certain function, critical function in biological system. But the question still remains the same. The general question is how living systems use energy to fight the second law of thermodynamics, right? So along these lines, we have worked on different problems, different specific problems in biology. Like, okay, so I'll give you some examples. But the central question for us is actually the following: you have the energy as energy cost. But you don't just waste it. You don't want to waste energy. What you really want to do is have certain performance of certain function, right? So what is the cost performance relation in these various different biological information processes? That's the question we try to answer in different biological systems. And the first system we actually worked on is actually a switch, very simple. So you have a biological switch, meaning that if you change some protein concentration, something else happens, right? So the functionality of the switch is characterized by this. This is input, this is output, by the slope, by the input out, the sharpness of the input-output relation. The sharper, the better in this case. So what we find is that actually for a non-equilibrium mechanism to work and for it to work well, namely to have a very high Hill coefficient, if you know what I mean, have a steeper response, it costs you more and more energy. For example, gamma, the energy dissipation is proportional to log of gamma. When gamma equals 1, that corresponds to no energy dissipation. So this is actually flat, no response at all. As gamma increases, this actually becomes sharper and sharper. In other words, this is using energy to build a more and more sensitive switch as a mechanism. I want to draw your attention to this is a work that we did more than 10 years ago, actually 12, was published 12 years ago, but work done was actually much earlier than that. It took so long to publish because nobody wants to accept this weird paper, and until it was Charlie came to the rescue. I don't know if you remember this, Charlie, but anyway, so it was published in PNAS 2008. That was the first our first work in this area, trying to relate energy dissipation, energy cost, and the performance of a certain biological function. And after that, we worked on sensory adaptation and how energy cost is related to how fast you can adapt to certain change of environment and how accurate you actually can adapt to, make yourself return to the set point that you are comfortable with. And then today, I'm going to tell you about the third example we worked in this direction, namely biochemical oscillations. Okay, so before I tell you all this, let me just review very, very briefly what you already heard from David's talk. So I'm very glad I followed David's talk. So but it's an important concept that you should remember is that in all equilibrium system, a thing called detail balance is satisfied. What do I mean by that is looking at the picture here, you have A, B, and C, three states, right? You can be the three states. In the equilibrium system, in the equilibrium steady state, then the claim is that the detail balance should be satisfied, meaning that the balance has to be satisfied between any pair of state, I and J, so that the rate of going from I to J should be exactly balanced the rate from going from J to I. So that's what detail balance is. That detail means that it's between any two microscopic states. Okay, so you have to be satisfied two microscopic states. For example, in this picture, it corresponds to K3 times probability in the C state is equal to exactly K minus 3 times PA here, right? Okay, so you can also look at that from a master equation point of view, which you also saw from David's talk. And Jij is the probability current from going from I to J, right? So detail balance basically corresponds to Jij is equal to Ji, right? So the current in two directions, in the reverse direction, are exactly the same. In a non-equilibrium system, this is not true anymore. So you can break this detail balance in any of the link that you have here, and the whole system will be out of equilibrium. So it's a very simple microscopic prescription of how you break detail balance. It's basically you change, you violate one of these relations, any link. I mean, the fact is, if you violate this in any link, all the other link will be violated too. Okay, so what is the consequence of that? The consequence of that is also you have heard is that the system is out of equilibrium. But remind you, out of equilibrium does not necessarily mean the system is always dynamic. It still can be in a steady state. So we are talking about in a non-equilibrium steady state. For example, if you look at a particular probability in particular state, after you reach a steady state, which means that this probability in that particular state doesn't change with time anymore. So if you measure just the probability of in the state C or A or B, you will not see any change, no dynamics there. But if you're able to measure the flux between A and C, or C and B, then in the non-equilibrium steady state, you will see a net flux. But in the equilibrium steady state, you will not see a net flux. Okay, so that's the difference between an equilibrium steady state and non-equilibrium steady state. So we're talking about steady state, and the difference is between equilibrium steady state and non-equilibrium steady state. Now, you have heard about entropy production rate. So this, in the non-equilibrium steady state, you have a entropy production rate. So this is remember, this is the rate. This is not a energy entropy production, amount of energy entropy production. This is a rate that is constantly being produced, right? And this goes like this. So it is basically on each link in this reaction, you have the forward flux minus the backward flux times the log of the ratio, basically the difference of their log. So you can easily prove this is always bigger or equal than 0, and it is only 0 when detail balance is satisfied. When Jij is equal to Jji, therefore, that is basically detail balance. Only when detail balance is satisfied, S dot equals 0. Anything going away from detail balance, violating detail balance, will lead to S dot bigger than 0. Okay. All right. So, I mean, this is not the invention of physicists, by the way. This is actually known, in very, very early on. And one of the pioneers in all of this is actually Terry Hill. I don't know how many people know who Terry Hill is. He, to me, is really the hero of this, unsung hero of this field. So, like, for example, in '77, he's already written a book about free energy transduction and biochemical cycle kinetics. So, in their language, it's about sort of the so-called thermodynamic forces. So he defined in this particular 3-node network, the thermodynamic force is basically KBT times log of gamma, right? What is this gamma? This is the gamma that I showed you before in the ultra-sensitivity switch. But the gamma is defined as such that if gamma is basically the product of all the rates, okay, in the clockwise direction, the product of all the rates, K1 times K2 times K3, divided by the product of all the rates in the counterclockwise direction, okay? K minus 1, K minus 2, K minus 3. The ratio of the product, the ratio of the two products is 1 if the system is in equilibrium, if the system has satisfied detail balance. If detail balance is broken, then gamma will not be 1. Will be either bigger than 1 or smaller than 1. Okay, and the current is basically the current that the cycle current is basically the current, the persistent current in the steady state that goes through the cycle, and the dissipation is basically J, the current times the force, right? So not surprising, the current times the force is the power consumption or the energy dissipation rate. It's a rate. All right, so okay, let's go to the simplicity that we try to use this kind of tools to try to understand biology, right? So the particular biological system I'm going to tell you today is oscillation, biochemical oscillation. It's seen everywhere in biology. You have the cell cycle. Here you have the p53 pathway oscillation. It's governed by the 2 key genes, p53, MDM2, plus a bunch of other things. But basically, if you observe some of the molecules, you see they go through time in a rhythmic way. And actually, in 2017, the Nobel Prize in Medicine was awarded for biological clock research, right, to these 3 gentlemen that discovered the key gene that's responsible for circadian clock. All right. So if you look at the circuit that gave rise to the biochemical circuit that gave rise to this oscillatory behavior in biological systems, you actually find that there are not too many of them. There are quite a few typical, there are only a few very typical motifs. And if you strip everything else down, you find that one of the most prominent network motif is the so-called activator inhibitor model, right? So you have R, which is the activator. It activates X and itself, and X is an inhibitor. It comes back to inhibit R with a time delay, right? And if you just look at that, if these parameters satisfy some certain condition, you see that they start to oscillate. You also have the rock, paper, and scissor model that C will suppress A, and A will suppress B, and B will suppress C. And if you look at the dynamics, if these parameters satisfy some condition, you also start to see oscillatory behavior with a phase lag of 60 degrees with each other, or 120 degrees rather, okay? And so on. So these 3 motifs that we studied, and we're not just interested in its dynamics, we are interested in the thermodynamics, meaning that we want to put noise into it. So these systems, because the finite number of molecules involved, as I said before, it's very, very noisy, right? It has a lot of noise. So even if you look at the deterministic equation, it oscillates with very nice, beautiful behavior. In a single cell, under biological conditions, when you observe these quantities, they actually oscillate, but they have a lot of noise on top of it. But we're not interested just in that. We're interested in the energetics. So where does energetics come in? I told you this before. in the previous slide that it's really the signature of equilibrium system is that a detailed balance is satisfied between any two pair of states. And in chemistry, this is called the thermodynamic relation. It should be called the equilibrium thermodynamic relation. Basically, gamma has to be equal to 1 for any cycles. And in biology, this cycle exists, and the detailed balance is broken, or the thermodynamic relation is violated because some of these reactions here, for example, here is coupled. So this is the driving force, outside driving force. To this gray area of the circuit is also coupled to the outside world, which is ATP hydrolysis. For example, here is this reaction, for example, is enhanced by coupling to a ATP hydrolysis reaction, release roughly around 20 kT of energy. Okay, so because of that, this is so therefore, I mean, of course, this is the open system because the fact that you are many of the reactions are coupled to things like ATP hydrolysis, which makes this whole system non-equilibrium, makes it more interesting, makes it the living system in the first place. Okay, all right. So let's look at the behavior of these systems. So we simulate. This is a simulation of such a system with finite number of particles involved. So this is a stochastic simulation of a system like this. You have different species of molecules. They interact with each other. We use Gillespie method to simulate the stochastic reaction between these different species of molecules. And because of the interaction satisfies certain condition, as I said before, these particular interaction or the network motif will give rise to oscillatory behavior. So we have done 500 different 500 independent runs of the simulation, because it's stochastic, not each one will look the same. And this is the raster plot of the peak time. When you have a peak time, we put a dot there, right? For trajectory 1, 2, 3, for all the 500. So these are the peak time. So if you look at, so we synchronize, we put them at the same phase at t equals 0. So when the first time when the peak happens after the first initial time, the distribution, which is the red line, is rather sharp, right? And the second peak is a little broadened, right? And the third and the statistics of the third and fourth and peak, so broaden and broaden and broaden. This is basically because the noise in the phase, right? The phase has a lot of fluctuation, even though it's still oscillating, but where it peaks will has fluctuates. And not only the fluctuates, because you have the system has time translation invariance. So the fluctuation mode, this mode of phase fluctuation, is a soft mode, which means that it satisfies diffusion equation. So it can be characterized by a diffusion constant. You can see this simply by looking at the variance of these peak distribution and look at versus the time you actually, which peak, this is the 50th peak and 100th peak, and look at the variance versus time. And you see the variance actually go linearly with time. You know that the phase is actually diffusing. It becomes fuzzier and fuzzier. By the end of the 100th oscillation, you can barely make out a very sharp peak time. Okay, so that's basically you are facing this. If you want to have a very clean oscillation, this is no good, right? Because you want the oscillation to be able to tell you the exact time after certain cycles, and it cannot. So the coherence time is basically inversely proportional to the phase diffusion constant. From here, you can define a phase diffusion constant. So that's basically what it is. And what we find is that, so long story short, is that the free energy can be used to suppress phase diffusion. So for all these different motifs, the repressilator motif network, the brusillator motif, the glycolysis motif network, we actually computed the diffusion, the phase diffusion constant versus the energy dissipation per period, right? I told you we can compute energy dissipation rate multiplied by the rate by the period of this oscillation. You get a energy dissipation amount. This is in unit of kT. And you see the diffusion, the phase diffusion goes down, right, with delta W. Meaning that in this circuit, energy is being used to suppress the phase diffusion, right? Okay, so that's basically what I'm trying to show in these figure, and in particular, it's actually able to suppress it with this following this inverse law, that diffusion constant equals it's saturated to a constant, but it goes like 1 over delta W minus WC. What delta W is the cost per period. Okay, why you have minus WC? Minus WC, it is actually the onset. So even for an infinite system, right? If your energy cost is not over, it's smaller than this onset energy, it will not have oscillation to start with. But for a finite system, the reason you want to spend ever more energy beyond the onset energy dissipation is to suppress the phase diffusion. Okay, so here's another plot. So we look at this network architecture, that we randomly pick parameters in this network, the rates and so on, and we look at the phase diffusion of those model versus the dissipation, and we see there's a lower bound. Basically, if you spend this much energy, you can never go below, the phase diffusion can never go below that. And this dotted line is exactly follow the expression that we shown before for a particular network architecture. Can we see any evidence for this? Well, I mean, so we talking to biologists and they say, well, we cannot do the experiment. That often happens when you talk to biologists. But then we look at the literature, we find that the simplest, so this is actually the most fun part, the simplest biochemical clock, circadian clock, is actually consists of only 3 molecules, chi A, chi B, chi C. And not only people know that, people can synthesize a clock in a test tube by extracting all these chi A, chi B, chi C and put them in the test tube, and they start to oscillate. No, that's not true. You have to add ATP. So you put these 3 molecules in certain proportion to each other in the test tube, and you put ATP in, and they start to oscillate. That's what they behave like. So what is oscillating? So not the amount of this molecule, but its modification. So what this means is that how much of it has been phosphorylated, right? So what is shown on the y-axis is the phosphorylation level of the chi C molecule, and it oscillates. Okay, so the experiment that we're able to find, which they did the experiment before, but not with the purpose of testing energetics, was the following. They try to understand how the quality of the oscillation depends on the ATP level. So they did experiment under different percentage of ATP, 100%. This percentage is versus the ADP, right? The rest is ADP. So they put in the solution different percentage of ATP and observe, they say, aha, look, different percentage of ATP up to 50% doesn't really change the period that much, right? The oscillation is still the same. And they say, oh, this clock is very robust against ATP-ADP ratio fluctuation. So that was the story, and they published in these papers. Then we say, well, we're not really interested in the period itself. We're interested in the quality, in the coherence time. So instead of just looking at the Fourier spectrum, let's look at the power spectrum and see the spread of the peak instead of where the peak is, right? Okay, then there we find something really interesting, is that if you look at the correlation of the time-delayed correlation, what we find is that as the ATP level goes down, the time-delayed correlation of this trajectory of these traces actually goes down. So here's what is shown, that so this is the inverse of the correlation time. Tau C is the correlation time, right? The coherence time. So this is better. Sorry, this is, did I make it wrong? Tau C is long. Yeah, okay, this is worse. This is better, right? So as ATP, sorry, this is good, this is worse. So this means that the tau C, the coherence time is, when the ATP level is low, the coherent time is short, right? So this curve goes like that. And this thick line corresponds to exactly the theoretical prediction. So from this experiment, the experiment seems to suggest that, yeah, our general relation seems to be correct, meaning that as more and more energy is being dissipated per period, depending on the ATP-ADP ratio, the quality of the clock become better in terms of having a longer coherence time when you have higher ratio of the ATP level. Okay, so that was the first story, and then from there we can figure out what is the good strategy to build a good biochemical clock. Well, first of all, if you imagine a clock is just the ticks on a circle, right? Then from each green dot corresponds to one particular biochemical state. Then you go from one state to the next, and so on. Right? But these are all chemical reactions. So if you don't do anything to it, it will be just a reversible process. So you wanna suppress the reverse reaction by couple it the forward reaction with the ATP hydrolysis, so it doesn't go backward. Right? So you suppress backward jumps by couple it to this energy consuming step, ATP hydrolysis. Well, that's okay. That's good. So even if you set it to 0, I mean, if you're 20 kT, you can certainly suppress it to 0. You still get a what's so-called the Poissonian clock, which means that each step is a Poisson step. So each step has a huge amount of noise in it, right? So it's not a periodic, because you go from here, the average have certain time, but your variance goes exactly the same as your average. So that's not a very good clock. So how do you deal with that? Well, to deal with that is that you put in more and more intermediate states before you finish the cycle, right? So because the Poissonian noise are independent of each other, you can, and the overall period is just the average of each individual stepping time. And because they are not correlated, you get 1 over square root of n gain. So to build a better clock is, first of all, you spend energy to suppress the reverse transition. And second, you actually building more and more ticks, intermediate states. So to actually average the Poissonian noise. All right, so I guess I have 8 minutes, 7 minutes. Okay. Okay, so the really that maybe 5 minutes, whatever the time I have left, I tell you another story, more recent story about coupled molecular oscillators, somewhat maybe related to what David was telling you about synchronous versus asynchronous one. So as I was telling you before, for one single clock, you can have a number of intermediate state for going to, let's say 24 hours is a cycle, and by putting dissipate energy, you can force the clock to go one direction, right? But it's a pretty noisy clock. And if you have two independent clocks, then they will pretty much go out of synchrony pretty easily, right? So if you imagine you have many of these individual clocks, you put in the soup, and if they don't talk to each other, they're pretty much out of sync. And as I said before, the clock is really because the number of molecules in a particular state is measured by the number of molecules in a particular state. Pretty soon, you have a uniform distribution around the cycle because there's no synchronization, right? So synchronization is critical. So how do you actually achieve synchronization? Do you need communication? So here is a picture of how you do communication in a chemical way, right? So you have two clocks, like phi 1 and phi 2 in a donut, right? So these two clocks communicate by swapping, by changing their phase. So because the phase is characterized by a physical object, meaning that the multiplication number of phosphate group on a molecule, you don't create or annihilate during the exchange process, during the communication steps. So you basically I will give some phosphate group to J, and J will receive that, or J will give some to I, so I will increase and J will decrease, right? So that's how you exchange the exchange reaction goes, and you put some rate omega here. And of course, once you introduce this reaction, you also create another loop. Besides the loop that one clock going into a cycle, you also create a local loop here, right? Local cycle. And this cycle also dissipates energy. And the amount of dissipation depends on the coupling strengths between these two. And also, I mean, as we later on show, also on the coupling frequency. How often do you do the exchange reaction? Actually, it turns out if under simple assumption of sort of a mean field assumption, if you assume every clock interacting with each other in the same way, right? Sort of a typical mean field approximation, you actually can solve this many particle Fokker-Planck equation exactly. And the solution is actually nothing but have the same form. I stress, even though it looks like Boltzmann distribution, is not Boltzmann distribution because the effective beta is different. So the beta is equal, the original beta, equilibrium beta is 1. Okay, so everything is scaled by kT. But this beta, this new beta, right, or this new temperature, beta minus 1, 1 inverse of beta is 1 plus k over omega. So meaning that it actually is hotter. This temperature is hotter. It is, although it is the same distribution as Boltzmann distribution, but the temperature is hotter. It's hotter because that you try to run the clock, right? And omega here is try to cool it down, try to be more synchronized. Okay, so that's the analogy. But the solution is actually, we didn't expect to have exact solution, but we did. It's sort of like the sort of approximation people usually typically use in many-body theory. Okay, so now we can study the synchronization as a phase transition, right? So the order parameter of the synchronization is basically the R. So what is R? R is if you add up all the different oscillator. This is the phase of oscillator J, e to the I phi j. If you average it, and if they are not synchronized, this will come to 0. So R will be 0. Okay. So R is the average oscillation, and in the asynchronous phase, R equals 0. In the synchronous phase, R is bigger than 0. And there's a phase transition between asynchronous state to synchronous state. And these two axes, this is the phase diagram, right? And the two axes basically shows you the interaction, the exchange interaction between two clocks. One is E0, the strength of the interaction. Another one is omega, it's the frequency of the interaction. How often do you do that? And it's normalized by K. What is K? K is the clock rate, the period of the clock. Basically tells you how often do you do the exchange during one period. Okay, and how strong you do it. What is this interaction strength? And we actually can exactly determine the critical line. The critical line is omega over omega plus K times E0 has to be equal to 2. When this thing is bigger than 2, R bigger than 0. It's in the synchronous phase. When this is less or equal than 2, this is R equals 0. This is the asynchronous phase. This is a second order phase transition here. Okay, then that's the phase transition. So that's the function, right? Synchronous to asynchronous. That's the function. What's the cost? The cost, the energy dissipation. We actually can compute energy dissipation in this many-body system by using, the Fokker-Planck equation. I won't go into the details, just a different way of expressing what the entropy production. Okay, and we find that there's an additional piece due to the coupling, due to the exchange reaction. Okay, we can plot that, and actually, when we plot that, it have this interesting feature. So how do we go about that? How do we try to understand it? We try to understand it by looking at, we cross it, right? We cross at each a line of a constant E0 or constant omega. What we find is that if we fix the interaction strands, we get a second order transition. For every individual E0, we get a second order transition with respect to energy. Okay, and also on top of that, we actually can have an envelope for this phase transition, meaning that for a given energy expenditure W, what is the maximum that you can achieve in terms of the synchronization. I won't go into the detail again. So this is the phase transition. We get the exponents, we get asymptotic behavior, and also this is the interesting part that in this two-dimensional control parameter space, omega is how frequent you do it and how strong you do exchange. You find there's an optimum design, meaning that yes, for a given energy expenditure, you can have a different combination of omega and E0 that give rise to the same energy cost. But in that combination, which is the best combination that give you the maximum synchronization. So this is the omega star and E star can also be determined. And it turns out it is much better to, instead of strengthen the interaction strengths, it is much better to actually make it more frequent. Okay, so maybe I don't know, I should stop or something, but this is the interesting part, which is we actually can connect back to experiment. So in experiment, people know that actually the Chi system, the system I told you before, so cyanobacteria can keep time, 24-hour cycle, have these two domains, C1 and C2. And C2 is the domain that have the two phosphate group that actually allows you to keep time, right? You have one, so you have total in the hexamer, you can have from 0 to 12 phosphate group. And that forms the 12 hours, by the way. Another, once you start to delete the 12 phosphate group, that's another 12 hours. It's actually 24 hours. It's actually exactly as we have in our watch. So, but you also have the C1 domain. That what it's doing there is nobody know really what it's doing there. And there's also a related puzzle is that, okay, so what about this individual hexamer? Do they actually talk to each other? And there's one very interesting experiment is that they actually can label a half of the hexamer, the molecule, by a tag. And later on, half of it, right? And they mix together. And later on, they measure it, and they see that actually all of it have this tag, which means they have already swapped partner after a certain time. And related puzzle is that, this is back to the title I have in the talk, is that, you know, if you just look at ATP hydrolysis of this phosphate group, this phosphorylation side, you would think that 2 ATP is new. Okay, I'm done. Okay, yeah, all right. So,
yeah, 2 minutes.
I have 2 minutes. Okay, so okay. So the puzzle is that if one monomer has 2 phosphorylation sites, each cycle will correspond to adding a phosphate group and then delete a phosphate group. Then you only cost you 2 ATP to run a clock per day, right? But people measure it, it takes 16 ATP. So that was the title of the talk. So here's what we come in. We think because the energy expenditure involved in exchanging between hexamers, right? So what happened, here's our story, right? Between 2 hexamers, what happened is that the exchange reaction happened because they actually disassemble and reassemble. In that disassemble or reassemble process, their phase has been averaged, right? So that's the exchange reaction. But that process costs you energy, and that's where the rest of the 14 ATP is cost, is to communicating, to facilitate the communication between, or the exchange reaction between 2 molecular clocks so that the whole system is synchronized. Okay, so that's basically the story, and you don't have to hear this this final summary. I just put it up here. Basically, what we're trying to do is to take the physics concept and tools, for example, non-equilibrium thermodynamics, to really try to understand how biological system works, how biological system try to use this energy to do various information processing tasks, and how well can they do, and how the performance of this function is limited by this energy expenditure through this information computation lens generally defined. Sorry, I ran a bit over time.
Okay, thank you.